Alcohol fermentation has been done for thousands of years, and in most cases it uses yeast known as Saccharomyces cerevisiae. In the production of wine, which again has been done for thousands of years, the extracted juice from grapes is treated with sulfur dioxide, a chemical that naturally kills occurring yeasts on the grapes themselves. There are some wines that use naturally occurring yeasts, but when you do this, the flavor of the wine can vary greatly, and many consumers don't like the tartness of the naturally occurring yeast. So today, most wine producers kill the naturally occurring yeast by using that chemical and then replace the yeast with known strains of Saccharomyces cerevisiae. These known strains are much more predictable in their flavor characteristics and profiles. Fermentation is allowed to proceed for a few days at fairly cool temperatures. This is followed by an aging process, which is carried out in wooden casks or sometimes stainless steel, and takes weeks, months, or even years to finish. During that process, flavors of the wine will change, partly as a result of the yeast metabolism and partly a result of the vats of oak or stainless steel that are used. Beers, on the other hand, are involving the fermentation of sugars from cereals, including barley, wheat, and rice. These grains are malted by being moistened and kept warm until the grains are partially germinated. That begins the enzymatic breakdown of starch into simple carbohydrates. The malt is then dried in an oven and partially charred or caramelized. This dried malt is then cracked grains, and they're steeped in hot water or mashed to extract the sugars and starches. The resulting liquid, called wort, is boiled to sterilize it. This stops enzymatic activity, kills organisms in that solution, and establishes flavor. Hops are also added, and these are dried flowers of a vine plant. After filtration and cooling of this wort, the yeast is added. The wort with the yeast is sealed off from oxygen in casks or barrels, and the yeast then ferments the sugars in the wort, creating ethyl alcohol and carbon dioxide. Stronger alcohols, such as brandy, whiskey, rum, vodka, and gin, are also produced using microbes. These distilled spirits begin with microbial production of alcohol, and then the alcohol is further concentrated by a chemical process known as distillation, where much of the alcohol is evaporated off, leaving a lot of the water behind. This can increase the alcohol concentration from roughly 10% up to 40 or 50%. The reason why this distillation process is required is because most microbes, such as yeast, end up dying in alcohol concentrations approaching 20%. So if an alcohol concentration over 20% is required, yeast can no longer be used. Your book talks a little bit about a characteristic or phenomenon seen in microorganisms and observed in the formation of some of these food items. This phenomenon is known as quorum sensing, and it is a way that microbes are able to communicate with each other in order to communicate information about the concentration of their relative population size. Quorum sensing uses single molecules, called inducer molecules, that are continuously released from the microbe into the environment, and other microbes of the same species can detect these inducer molecules. When these quorum-sensing signal molecules or inducer molecules are detected at a high enough concentration, the microbes then presume that there is a high concentration of them in that environment, and that they have reached quorum. Quorum would be the minimum concentration of these microbes to shift their behavior. The behavioral shifts that are governed by quorum sensing include biofilm formation, or adherence of bacteria to a solid surface, the production of pigmentation involved in photosynthesis, microbial protection, and survival, some infectious disease processes where microbes will not attack your body until there is a high enough concentration of them in your body to attack efficiently, and even the quality of some fermented foods that we've discussed in this section. There is a current trend among many people, especially in the Bay Area of California, to consume high quantities of fermented foods that are either raw fermented, naturally fermented, or even made at home. There's an increasing trend of making yogurts, kefirs, sauerkraut, pickles, kimchi, and bread at home in order to consume these fermented foods in higher concentrations. Some people are even engaged in producing fermented teas known as kombucha. All of this is a result of the concept that there are good bacteria and bad bacteria. And many of the good bacteria in our intestines include bacteria in the genera Lactobacillus and Bifidobacteria. The Lactobacillus species in particular are commonly found in many of these fermented food items. Foods supplemented with living organisms are referred to as probiotics. And probiotics can include these foods listed here, but can also include pills or tablets that can be purchased in pharmacies. Advocates claim that there is some scientific evidence indicating that probiotic foods can be helpful in the reduction of allergies, formation of a healthy digestive system, shortened duration of diarrheal episodes if you become ill, and other advantages. However, there is a lack of scientific evidence for most of these claims. And so the scientific community still considers probiotic use to be questionable. But if you enjoy these foods, why not eat them? There's also characteristics of some foods that contain what are referred to as prebiotics. Prebiotics are chemicals, not cells, like probiotics are. And these chemicals or molecules are particularly found in fermentable dietary fiber, such as resistant starch and inulin or fructose. And these molecules pass through the digestive system untouched by the human digestive properties and end up in your large intestine, where they supply the bacteria in your large intestine with excellent food options for the bacteria there. This promotes the growth of healthy or beneficial bacteria in your large intestine. And again, the idea is that this could be helpful for your overall digestive system and health. Just like with probiotics, the evidence of prebiotics is still lacking. But these are new areas of research, and as years progress, we get more and more scientific evidence, either in support of or against the use of probiotics for improving human health. At this point, we've covered the use of bacteria in our food, as well as the importance of bacteria in ecosystems on Earth. But humans haven't just stopped there. We've in fact started using bacteria in industrial processes, using biotechnology to generate products that we need today. Bacteria are of particular interest in these different fields because they are easy to grow, inexpensive, and reproduce rapidly. They are also easy targets of genetic engineering and can be easily altered in order to produce lots of different characteristics and products. In particular, genetic engineering, which uses recombinant DNA technology, can be used to achieve many goals. One of the most important of which of the 20th century leading into the 21st century was the Human Genome Project. The Human Genome Project began in the 1990s and was completed in the early 2000s and involved sequencing the DNA and understanding a little bit more about the 20,000 genes found in the human genome. Without microbes, none of this would have happened because the DNA needed to be pieced apart and separated into small sections, divided among microbes and sequenced that way. Another concept used in harnessing microbes in industry using genetic engineering is in gene therapy. Gene therapy is when a gene or DNA sequence is inserted into a bacterial cell or virus in order to treat a patient's genetic disease. In gene therapy, a malfunctioning gene is replaced with a correctly functioning copy of one. Gene therapy has been used in limited cases successfully to treat cancers, hemophilia, problems with a compromised immune system, and even some cases of blindness. A spin-off of gene therapy is a type of virotherapy that uses oncolytic viruses. Oncolytic viruses are viruses that seek out and destroy cancer cells. By training the viruses to specifically target only cancer cells, the viruses can invade the cancerous tissues of the body and destroy them, leaving the rest of the tissues intact. Microbes can also be used to produce chemicals that are needed by pharmaceutical companies for sick patients. Antibiotics, food additives, a variety of chemicals, cleaning products, enzymes, proteins, and carbohydrates are all examples of the different types of molecules being produced by genetic engineers in industry today. Important contributors to this, microbes have also played a role in the production of insulin by genetic engineering and also of vaccines. We're going to talk a little bit more about this and wrap up this chapter discussing bioremediation in the next and last section.